I've got HD in my family, and that's why most people think we give a donation to this conference, but if I'm being completely honest, it's actually so that I can get up here in front of this mic and just say something. When you don't have anything to say, you never get invited up here, and the only way to do it is you make a donation. And then they let you like introduce somebody who actually has something to say. But I'm here now, and I got the mic, so. Anyway, I get for real. I get the pleasure of introducing Dr. Jeff Carroll, which is pretty awesome. How many people in here know Dr. Jeff Carroll? I mean, I was given this bio here, but he doesn't need an introduction. No, I'm just kidding. So I was talking to him yesterday for a couple minutes, and I introduced myself and to the man who needs no introduction. And what he asked me to do was he really wanted me to embellish his accomplishments. And I was like, look, Dr. Carroll, I'm an entrepreneur. I don't just make stuff up and put it out there. Okay, I got a reputation to uphold, so I'm not going to do that. I'm just going to read it from the script here. So, Dr. Jeff Carroll, he first set foot on the moon in 1968. Okay, all right. Anyway, all jokes aside, personally, I would like to say thanks to Dr. Carroll. Thanks for what you do, and I really, really appreciate you taking the time to. Where are you? I'm trying to make eye contact with you. Oh, you're over there. Man, that makes way more sense. Yeah, thanks so much for what you do. I love reading on HD Buzz. All this, so much stuff comes out. It's mumbo jumbo, which I think is the technical term, and then you translate that into non-mumbo jumbo. For people like me, and that's huge, and I really appreciate that. So here's what I was actually told to say. So Dr. Jeff Carroll is a scientist studying HD as an associate professor at Western Washington University. He trained as a postdoctoral fellow in the lab of Marcy McDonald at Massachusetts General Hospital, Harvard Medical School, after completing his PhD under the supervision of Michael Hayden at UBC in Vancouver. His research is focused on understanding the links between metabolism and CAG expansion in the Huntington gene. As well as conducting research, Jeff is a member of an HD family and himself carries the mutation which causes the disease. Jeff also co-founded and serves as the co-editor-in-chief of HDBuzz.net with Dr. Ed Wilde. Please join me in welcoming Dr. Jeff Carroll.
Not for Canadians are bitter.
There's nothing.
Hi everybody, is my mic on? I promised I would stand still because apparently I'm annoying to record because I wander. I get bored. Hi everybody, it's really wonderful to be here. I'm really glad to be in the room. I was upstairs making my slides all day, so I'm sorry if I missed mingling with you. It was a real shame to be stuck up in the room, but I was supposed to do my presentation ahead of time, and for some mysterious reason I didn't. So I got to look out the lake and finish it, which was really nice. I also want to start by complaining. Some people have heard this already, I'm sorry to you guys, but I'm Canadian, as you know. Thank you. As I drove here today from where I live in Washington State, I was asked by the border guard, the Canadian border guard, what my citizenship was, and I said I'm a dual citizen. And she's like, "Where's your Canadian citizenship documents?" And I was like, "Well, I don't carry it with me. Like, I have my Nexus card." And she yelled at me. I got pulled in. I got yelled at. The other Canadian person was also very rude. So I'd like to file a complaint. If anyone has any connections, let me know. It's actually only because my wife tells me I'm not really Canadian, and it makes me sad every time. So to start off, to cut off the question that's going to come first, which is where's Ed? Dr. Wilde, fortunately, is in the running for a really huge grant, like like 2 million pounds, like a really big deal. In London, like in the UK, it's very like formal, the research process, and so he has to like get in uniform or whatever. I wear robes, or I'm picturing Hogwarts sort of, and he has to go in London to like a place and like talk to people for like hours, whether he gets the grant or not this week, and so he obviously had to do that and couldn't be here, and was really bummed about it. I go to a lot of meetings with Ed, and he this is going on the internet, right? Yeah. He talks all the time about how much he loves this community, enjoys this meeting, and is genuinely sorry not to be here with me. Also, he's funnier than me, so you're sorry. So this is the largest ever HSC conference, which is amazing. Someone told me there's over 300 people, is that right? This is incredible. And I'm highlighting it because it's gonna be my excuse for going over stuff that probably many of you who've seen me talk before have heard before, and I'm sorry to bore you to death. But I think it's really important that as these new people come in, they hear this stuff. And if people are only here for 3 days, they have to like learn everything they can and go back, hopefully enriched and empowered and excited, and tell their family. And so we're gonna hit some of these same messages over and over again, and I'll try to at least be somewhat entertaining. But I'm sorry for boring some of you who've seen all this before. I also have to apologize. That's my twins, Billy and Elijah. I should have had someone film them. We took them to Legoland. Wasn't that great, actually, but it was fun to hang out with kids. They're home with a sitter right now because I'm here with you guys, and so right after my talk, I'm grabbing my bag and hopping in the car to drive back to Bellingham. So I'm sorry to miss the dinner and dancing tonight. I came early for the white pad day and I couldn't swing both days, so there's my excuse for going home early tonight. So as Billy mentioned, I come from a Huntington's family, so for those of you who don't know who I am, that's me with my mom. Actually, it's me. Both of those pictures are me with my mom. One earlier than later, obviously. I'm also the one making the really smart, I'm going to be a professor someday face. That's me. Also, you can tell there's a seven. That paneling is amazing, and I wanted to my house now. That's my mom with 4 of her 6 kids. The 2 youngest, the ones we still call the little girls, were not yet born, or were doing something else that day. So my mom, Cindy, raised 6 of us. We didn't know at the time that she came from a family afflicted by Huntington's disease. We should have known, of course. The genetics are pretty simple. But my grandma was sort of like kind of this weird figure that nobody talked about, and then she was in a nursing home, and my mom stopped taking us to visit her, and just didn't talk about it. It seems weird now to say something like, we didn't talk about her grandma, because how can you not mention a person that's alive that's in your family, but we didn't talk about it. And it wasn't until I grew up and actually had moved away already that I found out my mom not only was at risk of Huntington's, and that was in fact what was wrong with my grandmother, but that my mother was starting to show symptoms. So I was around 20 years old. I just got married to my Canadian wife, which is why I'm Canadian for real. My dad, actually, I don't know why my dad told us, because anyway, that's a whole other story. But my dad sat my new wife and I down and said, this is happening, your mom has Huntington's disease. And my wife remembers, I don't remember, my wife remembers that I freaked out, like I stormed out of the room and I was incredibly upset. So obviously I knew on some level what that meant, although I would have told you that I didn't know that at the time, probably. My wife was like, what the hell is Huntington's disease? Because I never mentioned it to her because, of course, I thought we didn't have it. And so at the time I was in the Army, I was in the US Army, and so I didn't have a lot of time to process it or think about it, and didn't really do much productive for a little while. I got out of the Army in September of 2001, and I moved, because my wife had a house in Vancouver, we moved to Vancouver, and just got really lucky because Vancouver, as you all know, happens to be a huge center for Huntington's disease research, thanks to the pioneering efforts of Dr. Hayden, and now the other folks there, Dr. Raymond, Dr. Levitt, and others. I didn't know that. I was just lucky that I moved to this city that has some of the best HD research in the world. Also, it's Vancouver, so it was pretty great for me. And I started training, and so I did my bachelor's and my PhD there at UBC. During that time, I had the good fortune of working with Dr. Hayden, who taught me how to work on mouse models of Huntington's, which is what I do for my day job now. Actually, he didn't teach me. He doesn't put on gloves to go in the lab so much anymore. The people that he worked for Michael taught me how to work on mice. And now I also don't do it, and pay someone else to do it. It's the cycle of life. I write grants, actually, is what I do. That's my professional job. So I learned everything I know about HD in Michael's lab and from the people working there. It was an incredible experience. During the time I was in Michael's lab, I underwent pre-symptomatic testing myself. As soon as I found out about my mom, I knew that I had to find out what was going on. It took me a little while because just my life wasn't really in order. I was 20, I was traveling, I was in the military. It didn't really work out for a while. So once I got to Canada and got settled, I went through predictive testing at the clinic in UBC and eventually learned there that I had the same mutation that my mother had inherited. And so in the absence of effective treatments would develop the disease as well. This is my twins when they were cute. Cuter, love you guys. They're not gonna watch it talk of their dad. There's no chance they're gonna see this. This is actually the really funny thing about this picture is this is a picture of them. They're in a little tiny kiddie pool, like the ones you get like at Canadian Tire because I'm Canadian. The little 2-foot round inflatable bit, and we're on the 26th floor of a Yaletown condo, which is where we were living, so we set up that little pool out there. And so it's terrifying, because right behind them is like a 20-foot, I mean, they couldn't fall off and stuff. But, the point of this is not to tell you that story, the point of this is to tell you that for those of you who haven't heard me talk and don't know about this, Billy and Elijah were born after a genetic screening procedure called pre-implantation genetic diagnosis, or PGD. And it's an in vitro fertilization process where embryos are generated in a dish. I won't tell you how, because there's kids here. And when the embryos at a very early stage, like a little ball of cells, like 8 or 16 cells or something, one of the cells is removed by somebody with a really, I actually don't know what it looks like. I imagine it must be like a really specialized tool, like a little suction cup. And they take that one cell and they send it off to a lab in Detroit, actually, which is weird, but turns out it's hard to measure the DNA of one cell, and so there's specialized labs that do this. And they screen it and they say, okay, embryos number 1, 2, and 10, or whatever, have the mutation, the other ones don't. And so if you proceed with implanting the embryos that don't have HD, you have kids that have gone from 50% risk of HD to essentially 0, whatever the lab risk is. Of course, there's always some chance. And so that's an incredible thing, and I talk about this every time I talk about HD, and I'm sure a lot of you are sick of hearing me talk about it. But every time I do this, or every time I come to a meeting, someone comes up to me and says, my dad and my cousin heard about HD or PGD when you talked about it, and my cousin just had a baby and they're healthy, and I think that's one of the most impactful things that we can do in HD right now is for as many people as possible to consider PGD as an option when they decide to have kids. And I'll tell you, I come from a family where there are kids that are at risk and my kids that are not, and I know what that difference is. I know what it is to look at your kids and wonder, and for me and my wife to be able to look at our kids and not wonder is a lifelong blessing for us, as well as obviously for the kids. Although they probably don't care, actually. You don't really care about something you don't have. So this is my commercial, and I don't get paid by the PGD, Inc. What did I say? This rowdy table in the middle is laughing at me. I'm gonna go over here. Sorry, I'm moving. So do PGD. It's expensive. And unfortunately, it's not covered everywhere. Although increasingly more provinces are covering it now. I think, right? Is Ontario covering it? Yeah. BC. What site? Talk to someone who actually knows something about your provincial healthcare system, or get a loan, or sell your truck and do PGD. Always bothers me when I meet people that live in really nice houses and drive really nice cars. They're like, wow, $20,000 is a lot of money. It's like, what is not having HD worth, Okay, that's my last plug, and that's my introduction to myself. Sorry for those of you who know all that already. I wanted to talk, before we talk about the trials that have happened over the last year, a bit about some really cool basic science. Along the way, I became a nerdy scientist, and I'm super motivated by things that happen in the lab, just intrinsically. It's a really fun problem to work on. And it's been a pretty remarkable year for HD science, besides the obvious excitement about Huntington lowering, which I promise I'll talk about. So some basic science advances. So let's start with our history. Okay, so it's Huntington's disease because it was described by this guy, George Huntington, who only ever wrote one paper, and it was called On Chorea. And because he described in this paper Huntington's disease, we now call it Huntington's. So that was the massive first landmark of Huntington's was 1870, blah, 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 I can't remember, 1872, maybe, there we go, when this initial description of the disease, which at some point in the paper he says, gentlemen, because of course, no lady doctors, gentleman, he says, I call this to your attention as a medical curiosity only. So we're a medical curiosity, everyone. He's dead, so we win. Maybe he was a great guy, I don't know. I have no idea. Okay, so in 1872, the first time medicine knew anything about HD, right? Because you have to describe something before you can study it. And so people started studying it a little bit. There was a little bit of interest in HD around the early days of genetics. So HD was actually used by the scientists that came up with the word gene and genetics as an example of an autosomal dominant disease, meaning one in which you have a 50% chance of having it if your parents do. And so there was some basic science interest in HD, but not much therapeutic stuff happened. And then, of course, landmark paper in 1983, led by Jim Gussella, Nancy Wexler, and others, which found a region of chromosome 4 which was associated with the disease. And so those of you who are, I was going to say older, I don't know what to say that's less ominous. Those of you who are of more advanced age may remember an era when you could get what they called a linkage test, which was like a 95% chance or a 5% chance that you had HD, and that was based on this initial discovery where they didn't know the actual gene, but they knew like the location within the genome, and they could kind of look at it with these markers and say, yeah, you probably have it or you probably don't. It took 10 more years of incredibly hard work before a consortium of researchers, including many of those same people that were on the first paper, published this paper that actually identified the gene. And of course, we now call this the Huntington gene, just to make it confusing. And actually, just as a point of interest, in the paper it's called IT15. And I found out recently what this means. They were just like going down and manually looking at all the genes in this region of the chromosome and trying to figure out which one was associated with HD. And genes are called transcripts. And so this was interesting transcript number 15, because Marcy McDonald had like a list of genes she had to go through, and it was a really long list, and the 15th one was the HD gene, so she got lucky and didn't have to do the other like 100 or whatever. So that's history, there you go. So of course that changed everything, right? Because now we can say not just like you're likely to get HD, but you are or are not going to get HD with absolute certainty. And we need to have some nomenclature common here so we can talk about this. The Huntington's gene is found in our DNA, right, inside the nucleus of our cells, kind of really kept really safe and away from the rest of the machinery of the cell. And it's used as an instruction manual to sort of make a little molecular machine that helps your cells function called a protein. So all of the functional bits of our cells are mostly proteins, and they're basically encoded directly, not basically, they are encoded for directly at the genetic level. So the genetic information in your DNA is used to make proteins that make your cells function, and one of those is the Huntington protein. And if you ask me what the Huntington protein does, I'll say, I don't know. And that's what my whole lab is focused on. We know one bad thing it does, which is if you have this mutation, you're going to end up with Huntington's disease. The mutation, it's a little bit of a weird mutation. It's not like one letter of the DNA has changed. So scientists abbreviate the letters of DNA as A, T, G, and C, which are just symbols that we use to represent chemicals that are repeated in a sequence, and that sequence has information about how to build a protein. The Huntington's mutation turned out not to be a single mismatch or a single change, which can happen and cause disease, but rather an expansion of a long piece of sequence that is CAG, 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 CAG. And so for reasons we don't understand, this long CAG tract is found in people who don't have HD on average about 17 times in a row. There's 17 CAG, 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 CAGs. And if someone has HD, it's much longer than that, usually 40 or above. So this an elongation of this already long tract of CAGs causes the disease, and that leads to the creation of this protein. So this is a low-resolution picture of what the Huntington protein looked like. So in about 2014 or something, this was as much as we could like sort of see what one piece of the Huntington protein looked like, kind of this lumpy potato here. There's a normal copy, one that's not mutated, and scientists call that wild type. It just means like not the mutant one. So there's a normal Huntington protein in all of your cells, and there's also this mutant Huntington protein, which has a different shape, a slightly different shape, because of this long expansion of this repetitive region that was coded for by CAG. So I told you that genes are used as instructions to make proteins. That's true. I left out one step that you have to know. This is like this is like biology 101. So I just saved you tuition if you want to learn the one thing in biology 101. You actually have to remember this is it. It's called the central dogma of molecular biology. It was named that by Francis Crick, the co-discoverer of the DNA double helix. He said later that if he had known what the word dogma meant, he wouldn't have used it. Fun fact. because it means you can't question, and science is all about questioning. So the central dogma is that genetic information in our DNA is used to make proteins via this intermediate molecule in a closely related language to DNA. It's called RNA. It's a slightly different chemical, but it's the same idea, right? A sequence of sort of letters that has information about how to make a protein. This is the code that molecular biologists use to show protein sequence, not genetic sequence. This is the sequence of the Huntington protein. So when Marcy and all the other people discovered the Huntington gene, they knew it made a protein, and they found this. I'm showing you this just to prove to you how long this is. You're not supposed to get anything from this. The CAG's code for that long stretch of Q's, which is what we use to abbreviate amino acid glutamine, and then there's a whole humongous protein after it. And unfortunately for us, this protein didn't look like any other proteins that anyone knew about at the time. It was a very weird protein, very important. It's found in all kinds of species, all the way down to like jellyfish and slime molds have a Huntington protein, so do we. And so it's important. It does something our cells need, or some things that our cells need. But the sequence of the information told us who's going to get HD, but it didn't tell us like why. Like, what does this little machine do, and how does it go wrong to cause HD? I'll get that out of your way so you don't have to keep trying to figure it out. So, like I said, about 2014 or so, this was the best idea we had about what the Huntington protein looked like, and in 2018, we got this view. So, a group of researchers in Germany used a technique, a really amazing technique called cryo-EM microscopy, actually used by Rachel Harding, who gave a talk today you might have heard about, to look at the structure of the Huntington protein. So this is one individual Huntington protein made from a Huntington gene. There's a movie here. It's a little easier to see. I know you're not going to like figure out how to drug Huntington from looking at this, but it's just cool. So this is what the Huntington protein actually looks like. And so this is stunning. I never thought I would see this. I thought I would retire, we would hopefully cure HD, and I never thought that we would see this structure because the Huntington protein is too big, too weird, and too floppy. And so that's the technical term. It's too floppy. So I didn't think we'd be able to get a good structure of it. And because technology advanced like massively, all of a sudden now you guys can see this. Like I don't really cry. I'm dead inside. But when I saw this, like I literally got like kind of weepy. I mean, it's like I just never thought we'd be able to see it. Like this is the thing that causes the worst thing in my life. And now we have better ways of, once you know what something is, it's much easier to build tools, right, to interfere with it and to attach it to various things and mess around with it in the lab. So that happened this year. This is just, you don't need to know this stuff. We've always known for a long time that Huntington was important in part because it's found not just in your brain cells, but in every cell of your body. So for whatever it does for cells, it's so important that it's found in your skin cells, it's found in the cells of your lungs, it's found in your brain cells, which as we all know. And so that's, we've known that for a long time. This year and a bit, we also have learned something interesting about how important the Huntington protein is from humans. So in the lab, if you take a mouse genome and you bash out the Huntington gene, because that's how scientists figure out. Science is like going, you smack something and then you go, huh, you know, like, or you like poke it with a stick. So that's essentially what we do. So we take the mouse genome and we bash out the Huntington gene, because that's, you know, like, if you get rid of something, maybe you'll figure out what it was doing before. And unfortunately, it's not very useful because the mice are never born. So if you take away their Huntington gene, the normal Huntington gene, not a mutant one, the mice are never born. They absorb really, really early, and they never develop normally. And so we thought that meant this gene was important, obviously, for the process of development. And there's been the discovery of two families that have really weird mutations in their Huntington gene, not a CAG expansion, another kind of mutation that makes the protein go away. Basically, they basically don't have Huntington protein in their cells. These people, and they were discovered because the kids who have two copies of these genes that make no Huntington protein have really profound neurodevelopmental disorders. So they can't feed themselves, they can't do any self-care, and they're never getting any better. Their brains have really abnormal development. So this was really, I mean, this is sad for those families, but it's another piece of evidence that the Huntington gene does really important stuff. And we've had only lab stuff, and in the last year and a bit, we've learned from humans that this is also the case. A more uplifting and exciting advance this year has been what I think is really one of the most pivotal findings in Huntington's field in many years, which was the result of a work of a group called the GEM-HD Consortium. The GEM-HD Consortium. So GEM-HD is, again, another consortium of geneticists working on HD, many of the same people who cloned the gene. And they took data from patients that had participated in the PREDICT study and a similar study in Europe called the registry study. And they collected thousands and thousands of DNA samples. and they had the question, what modifies the age of onset of Huntington's disease? So obviously, we don't need to know what causes Huntington's disease genetically. We already know it's a CAG expansion, the Huntington gene. But they had a more clever question, and I put a slide in there somewhere. Always change your slides the day of the talk. It's great. I made it. This is what I was doing upstairs while you guys were having fun. Everybody's known this for a long time, since the gene was cloned. If you have a longer CAG, on average, you will get symptoms of HD earlier. So there's a line, like scientists like to draw nice straight lines. So there's a relationship between the age of onset and CAG size. And so unfortunately, kids that develop juvenile HD tend to have very long CAG repeats, and this holds true across the whole spectrum. But what scientists don't like is to show actual data. So I made this up, this isn't real data. But actually, maybe I should do that for my grant applications. It'd be so much easier. The actual data underlying these kinds of curves is a whole bunch of people, right? Individuals who had a CAG size of whatever, and they got sick at some age. And you take a bunch of those data points together, and you graph them on a graph like this, and you draw a line through, and the line is like the theoretical relationship. Each person is different, right? And what was known for a long time is that, like, let's say take those 3 points in red. What if those people all had really late onset, right, for their CAG size, and what if they were all from the same family? You might start thinking, maybe there's something genetic about the onset of HD, like that influences when your CAG tract is going to cause you to have symptoms. And so that's basically the hunch underlying this GEM HD consortium, which was a technique called GWAS, you're welcome, genome-wide association study, where they use this new technology of these, like, well, it's actually kind of established technology now, these like little microchips that read somebody's DNA at like 600,000 different places in their whole genome. So it's not like a whole genome sequence, but it's kind of like a high-level snapshot, like a Google Map view of the genome. And if you do that in enough people, you can ask questions like, where in the genome do people have differences that it's correlated with getting HD earlier or HD later? Does that make sense? So we know there's an environmental component, and we're not looking at that. This is just a genetic question. And what they found was that a number of genes have variations that if you inherit one version of this gene, you'll get HD earlier, and if you inherit another version, you might get it the opposite. Because I can't remember what I said. So there are genetic changes outside the HD gene that cause people to have HD earlier or later than they would be expected based on how long their CAG size is. And that wrench is there to remind me that almost all of these genes, very surprisingly, almost all of the genes that influence HD age of onset are genes that help cells repair their DNA. So there were a few scientists working on this idea that DNA repair. So DNA is obviously very important. If you screw it up, you get cancer and you die. So cells have we've all evolved like elaborate ways to keep our DNA healthy. We have a bunch of different genes that all work together to keep the genome intact. And one branch of this repair pathway just jumps out of this study and says that if you have variations in your DNA repair that make you less efficient, we think, at repairing DNA, you're likely to get HD earlier, and the opposite as well. There are individual variants that make people have onset as much as, like, say, 6 or 8 years later than they would be expected to. So if you don't take anything else from my long rambling talk about genome-wide association studies, take this. You don't have to wonder, is it possible to make HD happen later? Because now we know it is possible. There are humans walking around that have mutations in these genes that make them protected from HD onset for years in some cases. And so, of course, we can't take that person's gene and give it to everyone. That's not how it works. Or someday we'll be able to do that, but we can't do that now. But what we can do is use those genes as targets for drug companies to try to develop drugs that mimic that mutation that cause the same kind of effect. And drug companies are really good at that. That's what they do. And many of these genes, we know a lot about them. So it's like the opposite of the HD thing, because DNA repair is super important in cancer. And so if you're a drug company, you probably have somebody who knows an awful lot about the proteins that control or that regulate our DNA repair. So this is huge. It means we have now more targets. So obviously the Huntington gene itself is our main public enemy number one. Get rid of that. That causes the disease. But now we have all these other genes, like I think 10 now or more, that are also targets for making totally new approaches to Huntington if for some reason we can't mess with the Huntington protein itself, although I think we can. So this is huge. And it took the involvement of a lot of people. I hate this picture. This is a terrible picture of me. So I'll use it, because Ed uses it because it's a bad picture of me, so I'll just keep it. So just to give you a little flavor of like what we think this DNA repair thing is all about. The genes that are found on this genome-wide association study take part in a pathway in Huntington's. It's something called somatic instability. Instability of the body, soma, that's all that means. And the idea is that you take a ugly picture of someone like that, and that person inherits 42 CAGs, right, from their mom. And if you took a blood sample throughout their life and you measured their CAG size, it would be 42. But in certain tissues of the body, including parts of the brain, and in fact the parts of the brain that are most affected in HD, that CAG tract continues to get longer. So you're not going to pass it on because you don't make babies with your brain cells. I don't think. I don't know what you're into, but that seems weird. So it's not going to go anywhere generationally, but those individual cells are going to get a longer and longer CAG tract. So somatic, meaning cells of the body, instability, the CAG tract gets longer. It's very exciting because it's particularly bad in the part of the brain called the striatum. So that's not exciting, that's bad, but it means it might help us explain why that part of the brain is most affected by HD, because this process is really, really pronounced there. Also happens in the liver, which is weird. Which is why I try to cleanse my liver by poisoning it with alcohol. Okay, so that's the data. This was published now a bit over a year, even 2 years ago now. And this study has continued to grow, so they're adding more people to the study, and they're getting more and more resolution on what these variations are. This was my point to make a plug. So this is a plug for participating in Enroll HD. So now these other observational studies are done, and they've wrapped into this big meta-study called Enroll HD, which now has like 16, how many people? 16,000? 17,000? Huge collection of HD people, or people from HD families. You don't have to be tested, you don't have to know your CAG size, and they want family controls as well, so family members can sign up. You go in once a year. If you agree to, you give a DNA sample and other kinds of samples might be asked for. You fill out some questions. You do a little, let's say do a little dance. You don't do a dance, that would be weird. But your neurologist asks you awkward questions, like Dr. Levin asked me to think of all the words that start with F, and I was like, oh no. So it's just one gets stuck and you're like, I can't say it. So I did really bad on that part. It's not my fault. No, so I did enroll, it was great. I have to admit, I wasn't in enroll until recently. In my defense, I was in Predict for 10 years, I've done lumbar punctures, I've done a lot, but I was not in enroll, and so I was a hypocrite, you're right. And so I signed up for enroll this summer. I put a story on HD Buzz, you can go read it if you want to see what a visit is like. I would say it was fun, it wasn't, but it was fine. So this thing on the bottom has, you can see it's moving really funny. It's got this kind of diaphragm that's jumping. So one of the coolest new techniques in biology called CRISPR was used for the first time ever to make a genetically modified pig. And the first thing they did with this amazing new technology was use it to make a model of Huntington's. So I spent my whole life working on HD mouse models, and one thing that people like me will almost never tell you is our mice don't really look anything like HD patients. True story. Mice don't get chorea, right? So they move differently. So I can give a motor test and they move differently, but they don't have chorea. They don't have the most noticeable symptom of HD. And their brain, even the part of the brain that dies in HD, only shrinks a tiny bit. This new model this new pig model made by Jiajing Li at Emory and at a Chinese university with colleagues there was a huge advance. It actually moves a bit like an HD patient and has brain changes that look a lot more like an HD patient. So you might ask, why do you want a pig that looks like HD? Well, imagine you're Ionis or Roche or one of these companies developing a drug and you want to deliver it to the brains of HD patients and see if it makes their symptoms better. It's much better to do it A, in a large animal, right, that has a brain that's more like the size and shape of a human brain, and B, that has symptoms that actually look like the ones that happen in people that you're after treating. So that basic science advance, but I think a pretty huge one, although probably people didn't probably make on papers or anything. Okay, so that's a little snippet of the science that's been happening. The pace of science, I've been doing this now for longer than I want to say. It really feels very exponential right now. So the last HSC Therapeutics Conference in February, it was just every presentation was one of them was the structure of the protein. It was every presentation with some massive advance. And so it really feels really, really exciting in the labs. And I want to say that while we're going to talk about drug trials, don't forget that all those people in those labs are all still working, right? There's a ton happening. A lot of it funded by a nonprofit organization called CHDI Foundation. If you haven't heard of them before, they're a nonprofit focused only on developing therapies for HD. They don't fundraise, and so they don't talk a lot, so a lot of families don't know about them, but they're spending a lot of money, like $100 million a year kind of money, just on HD. And it's made a huge difference in the scale of work that's happening in labs around the world. Okay, so that's basic science. So let's talk a little bit about drug trials. So we had some sort of bad news this year. A couple trials didn't work as we'd hoped. So for example, there was a trial of a drug called laquinimod in a study called Legato HD. And we got updates on a lot of these in September, so just a few months ago at the European Huntington Disease Network. So you're going to see a lot of kind of bad pictures. Sorry, Ed took them, it's not my fault. But those are presentations shown by the drug companies, often for the first time, to the scientists and other people at EHDN. And so we saw, for example, that laquinimod missed its endpoint. It didn't do what we hoped it to, which is make people who have HD move better. So sometimes I've talked to people and they say, oh, this trial failed. The trial didn't fail, right? The trial was based on a good idea. This particular drug had a target of reducing a process called inflammation, sort of like the kind of process that happens when you get sick and you get a fever. A similar process happens inside your brain. It's a little separated from the one in the rest of your body, but you have an immune system in your brain. And in HD patients, it's like a little hyperactive. And this drug, laquinimod, is from MS, and it has the goal of reducing that inflammation to see if it could make the brain better and then make people's symptoms better. Very good idea, very good drug, didn't work. So this is not a failed trial, it's a negative trial. The drug doesn't do what it's supposed to do, but it was asked very well. The patient community rallied and volunteered and showed up, and the trial got run, and it didn't work. So now we can move on to the next thing. So I, like all of you, wish this had worked and everybody got better, but now at least we can use those people and resources for something that might work. So we don't have to take notes or anything. There's a story on HD Buzz about this. We had known for a little while that another trial of another drug run provided by Pfizer called a PDE10 inhibitor, doesn't matter, tested in a study called Amaryllis. We had known since 2016 that hadn't worked. Unfortunately, although again it was supported by a ton of data, it was a very reasonable trial to run. And at EHDN, Ralph Reilman gave us some more updates on that, so we're starting to see some more data. So much data gets collected from these trials, it takes a while just to process it, carefully. So we saw a little bit more of it at EHDN, including a somewhat surprising finding that the drug, which had made HD animal models a lot better and had looked really great, actually made people in the highest dose maybe a little worse. So this was their motor score, so their chorea might have actually gotten a little bit worse, and obviously wasn't the goal. And so to me, this is a good reminder. This is why you have to run trials. I mean, it's very frustrating sometimes, especially with the Huntington lowering stuff. People are like, let's just get into people. It's like weird things happen when you actually go in real human beings with a real drug, especially when there's a disease process going on. And unexpected things like, hey, you actually can make someone's chorea worse happen, and that's why we do trials, and it's why it's important to have placebo groups, yeah, again, kind of surprising. So as far as I know, the only trial with the drug being recruited in Canada right now is by this company, Vacinex. It's called the Signal trial. I just checked on the website. I think they're recruiting in Edmonton and Vancouver for cohort B, is that right, Lynn? So this trial is still ongoing. It's of an antibody that has a similar kind of goal, actually, of reducing inflammation, and some promising looking mouse work. And so this trial is ongoing and recruiting right now. And as far as I know, it's the only non-lowering trial still being run in Canada. Okay, does everyone want to talk about Huntington lowering? Somebody say something amusing because I have to drink water. From the rowdy table. I got beat really badly in ping pong last night. Turns out I'm terrible at it. That's why I'm never coming back to the Okanagan. Okay, so Huntington lowering, probably more important than ping pong skills. We're coming back to this central dogma of microbiology. And the idea here is that because the genetic because the information in our genes is not directly made into a protein, my remote's not working at all. I'll act it out, don't worry. DNA gets into RNA, becomes a protein. So the genetic information in our DNA is not directly, remember, used to make a protein. There's that kind of scratch copy made, and it's literally, well, it's partially to separate the kind of messy process of creating a protein, which happens in one part of the cell where your DNA is not. It's to allow your DNA to stay safe in the nucleus, not get messed with, where the protein making machinery is out in the kind of factory floor part of the cell where things are busy and messy. Oh, let's try it. Ooh. Okay. No, we just did it again. You guys are never going to know what's next. So I can't just tell you, which I will actually, if this doesn't work. It seems like every time I go on that one slide, it stops working. So Huntington lowering takes advantage of that intermediate copy, the mRNA, the messenger RNA, and tries to degrade it. Because the idea being, if you don't have any RNA, you don't have any protein, not just the idea, the fact, because we can do this in the lab pretty readily. And so there are a number of approaches focused on doing this to get rid of not all genes, obviously, but just the Huntington gene. We used to call it Huntington silencing or gene silencing. I try not to use that terminology anymore because it's not really accurate, because we're not really, silencing means turning off, right? And we're not doing that. We're lowering levels of the Huntington protein, and so nowadays we're trying to be like a little bit more specific and use this term Huntington lowering. So Huntington is both the gene and the protein that we're interested in lowering to try to affect Huntington's disease. The first, or the most advanced of these programs was started by a biotech company in Carlsbad, California called Ionis. They used to be called ISIS, but they're not called that anymore. It's not their fault. These are, never mind, I was going to make a head joke, that's not nice. Ionis started this program, so initially this drug was called Ionis, HTTRX, really rolls off the tongue. It has a new name now, so don't worry about that name because you can forget about it. Basically, these ASOs, drugs are called ASOs, antisense oligonucleotides, and all that is a short piece of DNA, basically, a single-stranded piece of DNA that has a heavily chemically modified backbone. And so it's not natural DNA, it's like Ionis has kind of hacked DNA in this case to get into cells and to be able to get where it needs to go to influence levels of a particular gene. So antisense oligonucleotides are themselves little tiny, about 20 base pairs of DNA that, again, has been modified in a certain way to allow it to have certain properties. And Ionis worked on this for decades. So this, in a cell with ASOs, the ASOs, this is exactly how it looks. This is an actual video of the process. It bounces in there on bouncy balls, and then it matches up. And because it's the DNA sequence itself, the ASO can go and bind to something, and what it matches and binds to is the Huntington RNA, the messenger that carries the genetic information from the DNA out to the protein-making machine. And when this happens, for reasons that I can tell you about later, well, I can't tell you about later, but I can tell you about someday, cells don't like this structure and they degrade it, they get rid of it. And so what you do is you get rid of the Huntington messenger RNA, and then no protein gets made. And so we've been able to do this in cells for years, and it works really well. And now we're finally getting to the point where we can do it in people. So that sort of, that's great, why haven't we fixed it? Well, one of the problems is the cells that we most need to get this to happen in are inside your skull. And people don't like it when you put needles through their skull very much. And we came up with this other way of delivering these drugs that actually is pretty cool. So the drug, the ASO, is just dissolved in sort of a saltwater solution, and it's injected at the base of the spine. And the fluid that's there is the same fluid that bathes your entire brain. And so if you dilute the drug into that fluid, it actually circulates, and it's kind of like, came up with this analogy I was so proud of yesterday. It's kind of like a garden sprayer, right? Like it goes all over your whole brain, your whole cortex. And so that little bit of drug injected down the base of the spine ends up like perfusing throughout your whole brain, just getting sprayed all over all of your brain tissue, which is great if you want to put an ASO and have it affect brain cells. The downside, of course, is that you have to get a spinal fluid injection, which is a bummer. Looking at the structure of these ASOs, it seems very unlikely to me that anyone's going to figure out a way to make them be taken as a pill. It's just your brain is very protected for a good reason, for molecules that look like DNA. And so for now, we're stuck with this access route of doing lumbar punctures. I've done a couple. Raise your hand if you've had a lumbar puncture. See, we all did it. It's fine. It's not that bad. Actually, my lumbar puncture, the worst part was the like little lidocaine injection that they give you, like a skin surface injection to numb it before they give you the needle, and that was the worst part. Actually, the worst part was that it was Dr. Levitt giving it to me. And so, like, those of you who've done it, you have to kind of like lean forward a little bit. And so I was like sitting on the edge of a bed, and I like had my head on the wall, and they grab your hips because they need to figure out which spinal section you're. And so Blair's grabbing my hips and like pushing on my back, and I'm like, "Don't tell anyone about this." I just told you about it. Actually, funny story, because I don't want to talk very much yet. Right before me was the first time, I think you had not been able to successfully do it without imaging for the first time in like 10 years or something on a patient that, let's just say there was stuff in the way. Okay, so it's fine is the point. The first trial I'm going to tell you about of this first drug from Ionis, and now partnered with the pharmaceutical company Roche. This first trial that was run was called a multiple ascending dose study. So what happens here, I mean, you're giving a novel drug to humans for the first time, so you have to be really careful. And you do that by giving people ascending doses. So you sort of have the first group start on a low dose, and you watch them really carefully, and then independent doctors review their health and say, yeah, okay, they look healthy. Let's go on to the next highest dose, and so on. And so you escalate the dose throughout the study until you're at the highest dose. The trial, the first safety trial that's completed, is called a phase 1/2A trial. It just means it's a safety study. The only point of a trial like this, the main outcome is safety. It's only 46 patients. This is not enough to see whether their HD symptoms got better or not. You need hundreds of patients to see that. It's just not enough people. It's on purpose. It's not an accident. It's on purpose because, again, you're giving people a novel drug. So you can start with 46 brave volunteers, enough that if something weird is going to happen, and rarely you'll see it, but few enough that you're not going to hurt people if there's unexpected side effects. You do it fast. So people were screened. They had 4 doses over 3 months, and then they were followed up for months, and actually months longer, and I'll tell you more later. So these 46 people, UK, Canada, Germany, they had active drug or placebo. You always have to have a placebo control, so a dummy treatment so that you can compare it and see how much effect the drug had. And we found out about the results of the safety study on December 11, 2017, which was a pretty exciting day because we woke up to news headlines like this. Does that just say the biggest breakthrough in science in 50 years? So it was exciting, right? Because what it showed, and to explain a little bit of the process here, these are what's called top-line results, and this is how this has to happen. The drug company puts out a basic summary of the findings as soon as they're available, and they have to, it's because of stock prices and blah, 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 they have to say as much as they're going to say at a certain time, and then there's actually tons of data that still needs to get analyzed, and actually the paper describing this, the actual scientific paper, is not out yet. So we only have a summary of the results, but it tells us, for what in this room we care about, the most important things. One, there were no serious adverse events. So nobody had horrible migraines or any other kind of acute problem that would have been worrying for the trial. Surprisingly to me, nobody dropped out of trial. So this is monthly spinal fluid injections in people who have HD, right? They have other things happening in their lives. And all 46 of these people stayed in a trial all the way to the end. So, huge kudos to them and their caregivers and family members for doing this incredible thing for the rest of us. More importantly, or even more importantly, maybe, every time these people were dosed with drug, the researchers took a little bit of their spinal fluid, right? So they're putting spinal fluid, they're putting the drug in the spinal fluid, and as they do that, they take off a little bit. And they can go and they can use this incredible new technique that's been developed that lets them measure levels of the Huntington protein, specifically the mutant Huntington protein. Remember the diagram we saw before? There's an incredible new lab technique that lets you sensitively measure how much mutant Huntington protein is in a sample, in this case in the spinal fluid. And Huntington's not normally supposed to be there, we don't think, but we think probably what happens as cells get sick and kind of start breaking down a little bit, little bits of them get kind of spit out into the spinal fluid, including the Huntington protein. And so this is really interesting because it lets researchers take that spinal fluid and measure the mutant Huntington in it and say, what happens to people who got drug? Well, this is what happens to people who got drug. This one did make me cry, one little tear. this graph is the trial, basically. This shows groups along the bottom, those are doses. So on the left is 0, that's people who got placebo, they didn't get any drug. And on the right, as you go up, people got more and more, a higher and higher dose. And what you can see, I was going to say on the y-axis, because I was going to lecture you guys like my undergrad, the dots are the measurements of the mutant Huntington protein in one person in each of those groups. And what you can see is that as you got more drug, you had less mutant Huntington in your spinal fluid. So we call this a dose-dependent lowering, meaning if you got more drug, you had less Huntington, which is exactly what you would expect from a drug whose job is to reduce levels of mutant Huntington. So this is incredibly exciting. And in fact, this might not even be as low as it can win because the people were still having reductions in their Huntington levels when the trial ended. So we don't know how low this can go, but this is a striking amount of reduction of the mutant Huntington protein. So again, we have no idea what this did to symptoms, right? This is not that kind of trial, but we know that it's safe enough that people showed up and got dosed and stayed in it, and we know that it reduced Huntington protein, which is incredibly exciting. And so this was first announced like kind of publicly at the HSC Therapeutics Conference. I'm sitting, of course, Ed cut me out. I'm sitting right there in the front row, about to cry, although I don't know it. Actually, Ed did not, nobody told me the results, so they really do keep their secrets very well. This was going to be a presentation by Ann Smith from Ionis and Sarah Tabrisi, the global clinical lead of this trial, sitting behind Ann there. The funny thing about this, though, is that Sarah ducked off to use the bathroom before the presentation because she was like nervous, and they announced her unexpectedly early, and she was not in the room for the biggest announcement like ever. And this is Ed texting her. So Professor Tabrisi missed the call. She eventually showed up. It was pretty amazing. So she gave a much more formal presentation of the results in another meeting in April at the American Academy of Neurology. So this is like a big deal, right? Other people in brain diseases are very interested in these results. And the take home, if you don't remember any of my rambling right now, so far the ASO is safe, well tolerated, and there was a dose-dependent reduction in Huntington, and people who got higher doses of drug had better effect. And so that's super exciting. So as part of that excitement, so small biotech companies tend not to run huge studies with hundreds of people because it's expensive. And so what typically happens in this situation is that a small biotech company like Ionis will partner with a large pharmaceutical company, and that's what happened here. So Ionis had partnered with a company which everywhere else is called Roche, but in North America is called Genentech, so it's a little confusing, just to keep you on your toes. And our friend HTTRX, that ASO that was safe and well tolerated, is now RG6042, which also rolls right off the tongue. This is like accounting, right? It's just what the people of the company call the drug. It's the same drug, it doesn't really matter. So Roche and Genentech are going to be the ones who actually run the next study. And we found out a little bit more about that more recently. So, we've got the so-called top-line results. We're still waiting. It won't be long, I think, before we see the full publication of all of the results sometime soon. We'll be able to talk more about it. You'll obviously hear about it in HD Buzz. And then there'll be what's called a pivotal trial. A pivotal study, that just means a study that's designed to show the drug actually influences symptoms, and therefore the drug should be approved and be able to give it to patients. So we were hoping that would start at the end of 2018 or the beginning of 2019, and now we have the answer to that. I'll tell you in a second. And one important thing, all the people that were in the phase 1 study stayed in it, basically. So Roche and Genentech, and I understand this cool thing called an open label extension. So, okay, 46 people is not enough to see whether their symptoms get better or not, but at least we can get some hints, right? And so what they did is instead of taking those people off the drug, they kept them on the drug. So I think, as far as I know, all 46 of those people are still getting the drug, and so they're sort of like the canaries in the coal mine, right, for whether we might see beneficial effects or even concerning effects to look for. And so that's great. It's sort of also kind of a reward for the people who are brave enough to be in that trial. Now they're on the drug ahead of time in case it works. Unfortunately, what it means is that you don't have a control group, right? Everybody in the safety study is now on the drug, and so in science you need a control group. You need someone to compare the people who got the drug to, and so Roche has come up with this idea of having they call it a natural history study. So they're going to take, I think, 100 HD patients that kind of match the people that were in the phase 1 study, and they're going to recruit them and kind of, it's not really a control group because they weren't in the same study, but at least it lets them kind of say, okay, if we measure these people in the exact same way, can we see a difference between people who were treated on the drug and people who weren't, who all have HD. So the most recent update happened just very recently on September 15th. This is Scott Schobel from Roche giving an update about the pivotal study at the EHDN meeting. These are his slides that I meant to put underneath of them. So I just stole this from him. Sorry, Roche people. You can actually get these slides, they're public. You can go to the HSC Buzz story and you can click through, so you need to try to scroll all this down. This was the announcement that Scott made in Vienna. The safety study has been completed. That's the technical details about the study. You don't need to worry about that stuff. All those people are now on this so-called open label. So those people have been transitioned into getting drug. All of them are. And they're going to have this matched group of 100 people that are not on drug as a sort of a control group for those, this natural history study that's going to be launching. And then, of course, the one everyone wants to talk about, the so-called pivotal study. And so we now know what this is going to look like. It's going to be 660 patients. That's a lot. It's going to be people that have manifest HD, and they're going to be treated for 25 months plus follow-up. And the goal of this is not just to see whether it's safe and well tolerated. We hope it continues to be that. The goal of this is to see, does this make their HD better? So the inclusion criteria are here. So this is the characteristics of people. Words are hard. The characteristics of people have to be in this trial. So they have to have clinically diagnosed Huntington's disease. So this is not a trial in pre-symptomatic people yet. They have to be between age 25 and 65 years. There's a score called the CAP score. It's a bit strange. It's just like a mathematical way to combine your CAG size and your age. And so from trials like PREDICT and TRACK, we know that if you limit people in the trial to a narrow range on this CAP, not a narrow range, but within a range on this with this CAP score, you get kind of cleaner data and you can see changes more easily. People have to be fairly independent and ambulatory, meaning they can walk, and verbal, so they can participate in some of the tests. And again, the 660 people are going to be seen in 80 or 90 different clinical sites in 15 different countries. It's a huge worldwide study. We, of course, expect there to be sites in Canada. And what they're going to be done is divided into 3 different groups. No one's going to know who's in what group until the end of the trial. One of the groups will be a placebo, right, a control group. One of them will get the highest dose that was tested in the safety study every month, the same way that the first study was done. The second group is really interesting, I think. Roche decided to take another group of people, so another whole chunk of people, and give them the drug every month initially, and then every other month, alternating with a dummy treatment. So those people effectively will be getting dosed with the ASO every 2 months instead of every 1 month. I mean, the point of that must be to let us know if let's say this drug works and everything that we hope for comes true and the drug works, and if the people in the every other month treatment group do as well as the people in the every month, now that's justification for going forward with a plan where we give people the drug every other month instead of every month, right? I would get a spinal injection every day if it would cure HD, but every other month is a lot easier to fit into your life, particularly if you live somewhere far away from a center that can give you this kind of drug. So that's the design of the trial. I think it's very smart. I think Roche made a good decision in designing it this way. I think they're putting their money where their mouth is, because it's expensive to put another 220 people on that treatment regimen, but if it works, it's going to make the ultimate treatment that comes from it, I think, a lot easier to give to people. So, sorry, I'm supposed to be watching time. So that's huge. That's like the most exciting trial to ever happen since I've been studying HD, but it's not all that's happening. There's other trials happening. There's a company called Wave Life Sciences. Some of their there's a couple people from the company here. And Wave they also use ASOs of a similar chemistry to Ionis. And they're doing a Huntington trial as well. In fact, they're doing more than one Huntington trial right now. They're running 2 separate trials that some of you might have heard Dr. Gutman talk about today. So Wave's approach is a little different than the one taken by Roche and Ionis. It's fundamentally the same thing. It's using ASOs to try to interfere with this process of turning a gene into a protein. But Wave Life Science, their approach targets little tiny genetic variations in the Huntington gene, not the CAG tract itself, for reasons that will bore you to tears. Come see me later. You can't target the CAG tract itself with ASOs. It's too long. You would basically knock down other genes because there's other genes that have those CAGs. And so the wave ASOs target little tiny variations, right? Sort of meaningless but there variations that occur in all of our genes, which is why we're not identical twins. Wave takes advantage of the fact that some of these little variations are on the copy of the Huntington gene that it has a mutation, the long CAG tract. And so by targeting that, they're able to only reduce the mutant Huntington protein and not the normal Huntington protein. And so, in theory, that's better. Like, if this is an important gene, maybe we should just leave the one copy that's normal. So on the surface, it seems like the better way to go. The downside, there's always downside. The downside is that you have to have not only the HD mutation, this long CAG tract, you also have to have this other little variation that's actually the target of the ASO. And so I think about half of HD patients are thought to maybe have one of these variants, and there's one trial targeting that. And then the Wave has a second trial running in parallel with another variant that about half of the remaining people might be eligible for. So upsides and downsides of that approach, it's incredibly exciting if you think about it that we went from a time when we didn't have any exciting Huntington lowering trials to now we actually have like competing programs, right? Like Ionis and Roche are driving forward as fast as they can with an approach that targets both copies. Wave is running 2 and maybe eventually 3 different versions of an ASO that target only the mutant Huntington protein, and nobody knows which one's going to be better, safer, whatever, right? This is, but we're not going to have to like sequentially start the next trial if the Roche Ionis trial doesn't end up with the result we want. These trials are happening in parallel, which I think is incredible. A really good testament to this community. There's other trials happening, so it's not just ASOs out there, although I tend to focus on them because I'm interested in them and they're the farthest advanced trials right now. This just came out, again I stole this poster online and made a slide out of it. This came out in October. This is from, data from a company called Voyager. Voyager uses, instead of regular injections of a little bit of DNA that works like an ASO, Voyager takes another approach where they sort of take the instructions for making that kind of ASO and they package it into a virus. And then you can take that virus and put it into the brain cells, and the brain cells themselves will start to make the thing that reduces the levels of the Huntington protein. You sort of turn them into little drug factories. So, upside, you only need one injection. You're asleep for it because they're stick a needle deep in your brain, and you get one injection of a virus, and your brain cells are forever making that thing and reducing levels of the Huntington protein. Downside, you can't turn it off. So if later we find out that there's difficult consequences of this, unlike ASOs where you could just stop giving them to people and they would go away, with this kind of viral delivery or what we call gene therapy, like once it's there, it's there really for the life of the cell, just the life of us. I have to be honest, I have been a little bit like, this has always been kind of second string to me. It didn't seem like it was ready for prime time. This data is really remarkable. I won't go into the details, but Voyager have now published, these are from primates, these are from monkeys, not like little mouse or rats, and they've got really remarkable reductions of Huntington protein after, again, just a series of one injection done at one time during surgery. So there are other approaches besides just the ASOs that are really approaching the clinic, and Voyager is not. I mean, they're really gunning towards going into the clinic pretty soon, it seems like, based on these data. And then another thing that came out at the European Huntington's Disease Network meeting kind of came out of the blue. This was not on my radar until this summer, basically, to be honest. A company called PTC Therapeutics in New Jersey has a drug. Scientists call it a small molecule. It just means like aspirin, something you can take as a pill. Not an ASO, you don't have to inject it into the spinal fluid. And this is from, very important to say, this is mouse data. So in mice, just preface everything with in mice fed with this drug, basically given the drinking water to a mouse, levels of the Huntington protein in their brain went down by about like 75 or 80%. So using an orally dosed pill, basically, they were able to reduce levels of Huntington protein in the brains of an animal by like 75 or 80%. That's remarkable. If you told me this drug would work and how it works, I would have not believed you. But it definitely does, at least in these animal models that have been done. And so there's a long way to go, right? We got to figure out, can they make it selective? Is it safe? Can you lower Huntington over the body without having problems? But this is also rapidly advancing, and PTC said at the European Huntington's Disease Network meeting they wanted to be in people in 2020, so not a long time away from wanting to go into human trials. And beyond that, we live in this weird world now where it's actually possible to manipulate DNA, right? To do the thing that central dogma says you can't do and change a genetic sequence using technologies like CRISPR, Cas9, and other kinds of what we call genome engineering techniques. It's possible to go in and actually manipulate DNA, and in theory it's possible now to go and remove, say, the CAG track, leave the HD gene intact, and just do a little bit of surgery and take out the extra CAGs. Now, again, it seemed like science fiction until someone did it. Now we do it in the lab routinely. I do some mice in my lab all the time. There are companies doing this for other diseases. I'm sure Huntington's will be on people's radar as something to try. So this is, a stage further back. It's still very much just at the lab level now. But years from now, it could be possible to actually manipulate DNA and basically fix the underlying cause of HD rather than just trying to cope with it. And this, believe it or not, that's not even like an exhaustive list of all the companies trying to do Huntington lowering. There's a company called Shire that's doing zinc finger approaches that actually block the DNA from being transcribed into RNA. There's another company, Unicure, working on another viral gene therapy approach. So there are literally more companies trying to lower Huntington in humans than I have time to tell you about, which is a pretty incredible state of affairs. So what can you do now? None of us can get any of these drugs. We don't know if they work yet. So what can we do in the meantime? This is a really weirdly inspirational slide. It's like something, I haven't been in church in a long time, but I feel like that's the kind of thing that they would show you in church. Like they're going to be inspirational quote up next. There is, actually. Okay, so one, look after yourself. We've been talking at this meeting in a number of venues about the fact that people who are healthy generally do better in HD, even if you have HD. And so if you ask, what's the best thing I can do right now? Eat well, sleep well, do all the things your doctor tells you to do that I don't do. Don't be like me. Sign up for Enroll HD, right? Things like that Gem HD, that massive advance, only happened because thousands and thousands of HD families participated, donated their DNA samples, consented to have research happen, and we have no idea if these Huntington lowering drugs are going to work, or if we need the next layer of things that'll be empowered by people participating in HD. And stay informed. All the companies involved with this stuff have been chatting with HD Buzz. Both Roche and Ionis and Wave have been great about giving us updates. So you don't need to agonize over when will we know. Just subscribe to HD Buzz's email feature, and we'll send you updates when they happen, and we'll know as soon as anyone does. I don't know why I'm showing you the Enroll HD website, you don't know how to Google things. If you had Googled Enroll HD, that's what you would have found. There's a website there, and you can find sites near you, including a bunch in Canada. Finally, stay informed. So watch HD Buzz for updates about these Huntington lowering trials and other really exciting stuff happening in the future. And with that, I will say thank you. There's two of us usually. It's a lot. I feel I'm sorry, it's a lot, but it's a lot of information. And so I thought I would just power through it and then tell you guys that I'm happy to take questions. And I am. So I think there's microphones, right, that people can ask questions. So if you have questions, come to the mic. I would say to tolerate me, but don't, because apparently this is being recorded. So I answered all of your questions.
Hey, John.
Hi.
How's it going?
Great.
We had a question on Facebook. If you could just talk about what non-Huntington lowering means.
Non-Huntington lowering. Oh, so I think in my mind, the kind of trials that are being run right now sort of fall into two kind of bins, and one of them is these Huntington lowering approaches, everything from ASOs to zinc fingers to all those different zoo technologies I talked about, and then the other things focused on other kinds of drugs I kind of call non-Huntington lowering, just sort of a distinction that's useful for me. Hi.
Hi. You have a cap on age, like from 25 to 65, Why isn't it higher?
I don't have any caps because I don't work for Roche, but you know, to be honest, I'm not sure. Does any of the physicians want to maybe potentially address why we want to have a cap of 65? Or Roche?
It's not ageism, first of all. It's the same reason that all of these inclusion-exclusion criteria are determined why there's a cap score. This has to be the most efficient trial possible. And in order to do that, we have to use the best measures we have. We have to select the ideal patient population. And what we're trying to do is decrease variability. When we're looking at measures, we want to have something that's sensitive and we want to have limited variability. That gives us the best chance of seeing differences, even if they're small. And if we didn't do that, and we just willy-nilly did a trial, the chances are the variability would be so great we'd never be able to see anything. So I didn't choose 65. It's all done based on the statistics, the observational studies, and choosing the best group of patients that are getting, not best, it's not a judgment call on who's a better patient who isn't. It's which group of individuals, based on what we understand about the disease, is going to give us the greatest chance of seeing if there is a real difference or not. And it just turns out that the numbers work out this way. If it was a 10,000 person study, and we had the money to do that, then we could have a broader, it's a matter of narrowing down the criteria so you can use a reasonable number of individuals and see a difference. That's the best answer I can give.
That's okay. Yeah, and I would add one more thing, which is I go through this exact same thing in my head. I wonder about people in my family and whether they'd be eligible, and I immediately start going down the same train of thought that all of you are going down, right? But ultimately, the thing it's like a mantra, you have to just remember it, is trials are not treatment. Like, the goal of this trial is not to treat the 660 people that are in the trial. The point of this trial is to figure out whether this works for everyone. And so we want this trial to run as efficiently and quickly as possible. And you know, the powers that be have sort of done the math and decided that this is the best and most efficient way. And it's not, you know, the goal again is not to treat the people that are in this trial, it's to treat everybody who needs it, hopefully soon. Hello, stranger.
Hello there. I had a question. I feel like I should probably know this, but about the Voyager technology. You said that they were able to lower the amount of Huntington in primates, right? Were those primates, were they lowering mutant Huntington? Were those primates an HD model? And if so, did they see anything about improvement of symptoms in those primates?
So I'm a little nervous for talking about somebody else's data, but from what I read of it, they're lower than normal Huntington in a primate that doesn't have mutant HD.
Okay, so it wasn't an HD model, it was just lowering the normal gene just to, I guess, look for safety and see if it could be done.
Yeah, okay. And to see, like, you know, they're relying on the virus to spread enough to cover enough brain tissue to have an impact, and they were just trying to see technically if they could do that, and it seems pretty amazing. Hi. You need a shorter mic.
I had more of a general question because, I mean, there are so many exciting trials coming up, and all of us want to get involved. So what happens if somebody's getting involved in one of the trials and then somehow it ends? And then can this person participate in another one? Like, that's the sort of general criteria of participating more than one.
Yeah, so for some of these, like the gene therapy one that I mentioned, it may be possible that in future that those people wouldn't be able to participate, but generally that's not the case. I think that's fair to say. I'm gonna let a real doctor maybe answer that. In the meantime, I will do a dance. No, I won't. And you don't want to see that either.
So each of the trials has specific inclusion and exclusion criteria, and there are many people in the room that have done a series of studies. One study finishes, another study starts. But in these antisense oligonucleotide studies, the disclosing anything that I'm not supposed to disclose, it may be a problem.
to switch from one study to another. However, we're very lucky that there should be no good reason to do that, because if you're in the current studies or the ones that are being designed and starting soon, there's the option to continue in the study with what's called an open label extension. So the patients can continue, or the individuals can continue after the study's finished in that study. So there should be no real reason to switch from Wave to Roche or the other way around, because they will be eligible to continue until there's a reason not to continue. Either the drug is made approved and you can get it through the regular ways, or there's a problem and they stop the process.
Jeff, did you have another online question? Yeah. I just realized how weird it is that a bunch of people are watching this right now.
It's pretty cool. It's been great. It's been great all weekend. This one's via text message, and it's, if one of the trials are successful, is there a chance that it could be fast-tracked?
Is it possible it could be fast-tracked? So, okay, I'm going to put on my In My Wildest Dreams hat, okay? So, this is all, take this all with a grain of salt. We have an example from another neurological disease called spinal muscular atrophy, or SMA, in which there was another ASO being tested for this other genetic disease, a neuromuscular disease of children. In particular, kids in this trial were really sick, and in fact, had they not been treated, the course of the disease is that they would eventually die. And they were given an ASO. It works a little differently than the one that we're testing, but it's the same basic principle. And the trial was actually halted early by the regulatory authorities because the drug worked so well. The kids were staying alive that had the drug, and the kids in the placebo unfortunately weren't, and so it was felt that it was unethical to keep the trial going. Now, that's a little different than HD, and that drug actually got approved really fast, so it's possible. So if you ask me, is it possible? Yes, it could happen. HD is a different animal. As we all know, HD's progression is slow and variable, and there's good days and bad days, and I would be very surprised if the results of any of these trials were so amazing and obvious that something like that happened in HD. Right? It's inherently harder to see a really improvement and a really slow progression compared to like a healthy baby or one who gets really, really sick. But it's possible.
This one's related to the Roche trial. Enrollment is expected to begin in 2019. Will the enrollment be staggered? And how long will the total time for the study be?
Yeah, I can't speak specifically for Roche. I know that they were hoping to launch the trial at the end of basically this month or next month formally, and then start enrolling people very early next year. You can look at other multi-center trials, like not all the centers are ready on day one. So I'm not going to say a time limit because I know that's what everybody wants, but any guess I give you will be completely a guess. So fundamentally, the sites have to get up and running, they have to enroll, and then each, the last patient has to go through 25 months of treatment before the trial's over. So it will be longer than 25 months. How much longer, I don't know. And we can all help by enrolling and participating as fast as we can.
This one's related to lumbar punctures. Are there any concerns with safety with patients with chorea?
Oh, that's a good question. Blair, do you see problems with? Is it hard with chorea? Does it make it more difficult?
Certainly, it can, a lot of chorea can make doing any procedure more difficult. We've had experience both at all of the sites that are involved in both studies with individuals with chorea, and we've been able to do it. Other aspects are actually more important, and there's limits on body habitus and BMI that are included that make a bigger difference. So it can make it a little harder, but it's not. It doesn't. We're not limiting individuals and the amount of chorea they had, and it can be done in individuals with a lot of chorea. So yes, it makes it a little harder, but it doesn't prevent it from happening.
You guys are good at your job. That's amazing. This is why I work on mice, because they hold still. It's like Jeff, was there another question? Oh.
I was just wondering if there are any studies active right now for folks in very late stage Huntington's. Are there any studies or any drug trials for people that are already kind of dubbed beyond repair?
I guess I don't think of it as beyond repair. The Vaccinax trial is still running. I'm not sure what their cap is. The Roche trial by design wouldn't include somebody in the late stages. And part of that is just that it's very difficult with people in that situation to measure their symptoms. Or, for example, to give them a thinking test, a cognitive test, if they have a really hard time communicating. And so it's not so much that people don't want to treat people in late stages, it's just very difficult for them to participate in a way. And I'll say one thing too, just thinking about it, with these Huntington lowering things, if we had the capacity, let's say I wave a magic wand and one of those drugs works and we're able to arrest somebody's symptoms, at some point you have to ask yourself, ethically, where would you, at what stage would you not want to be preserved in that stage for a really, really long time? It's a very difficult thought. Because some of these things, even in our wildest dreams, might arrest progression of disease, but they may not be able to give people back what they had. So that's my non-physician answer.
So just to let people know, next week's meeting, I'm participating in the steering committee or international oversight committee for Enroll. So thanks for giving the plug for Enroll. There's now 17,500 patients. We're projecting 20,000 by the end of next year. So for people that have been involved in Enroll, this is fantastic. We've had people that are 5 years into the study. The longitudinal aspects of the study are unbelievable. But to answer this kind of question, it's hard for people with advanced Huntington's to participate. So what we're planning, and my homework for next week's meeting is there's a whole protocol being developed for people with advanced Huntington's to be part of an easier protocol called Enroll Light. So people that can't do the cognitive testing, filling out all the questionnaires, we still want to capture information and have information in the database about advanced people, but it's going to be a lot easier for them. And the hope is that further recruitment within Enroll will be in sort of a steady state with people that are not continuing to participate. And it's amazing, we have, I think, about 10% dropout per year, which in a huge study is unbelievable. That's fantastic when you're trying to design clinical trials. So we're trying to bring in the young people, the pre-symptomatic individuals, the early symptomatic individuals, so we'll have sort of a refreshing of the people that will go into this Enroll Light, so we'll have a continuous stream for a long time to come, refreshing the group. It's interesting, in Canada we're very lucky that we have many sites involved. There's now a struggle because there's many countries that never got up and running, and CHDI is spending $35 million a year just on Enroll. It's costing about $10 million a year just to keep the biosamples in storage. It's unbelievable amounts of money to continue going. So there are some countries that never got up and running, and they're saying, what's the marginal utility? How much more effective is it going to be to have 22,000 than 20,000? We got to say, okay, let's be practical here. So we've tried our best to go from coast to coast. We have new sites in Canada and Halifax that have just been put on, and in Ottawa that's just been put on. In addition to Vancouver, Calgary, Edmonton, Toronto, now Ottawa, Montreal, Halifax. So we've done a good job covering from coast to coast, I think, and we're trying to do that with the other studies that are up and running.
Sometimes things like this Enroll trial can seem a little bit obscure and it's hard to picture what good they are, but think about that genome-wide association study. And another thing I'll tell you is when we meet new drug companies, people that are going to maybe have an HD program, when they hear about this incredible resource of data from patients, it's like the drug companies love it, and they say this is great, this is exactly what we need. So we're really building the platform that we need to do all the trials that we're going to get run. Is there any last questions? I think I'm almost out of time. Okay, great question.
Just if you could clarify, when you were saying with the gene lowering, if it gets lower, will that improve the the symptoms of the person, or would just stay at where they are at the time that they're lower and not get worse?
That's the $1 million question. Well, it's much more than $1 million question. That's the insert big number here question. So I'll tell you that as a scientist, I have reasons that I'm a little bit optimistic that we could maybe actually have some improvement, which come from animal studies, where the benefits of even a short time of Huntington lowering were much more protracted, lasted for many more months than we would have thought. And in another set of animal experiments, animals that had mutant Huntington completely shut off in a way that we can't do in humans, actually improved in their symptoms, they didn't just stay. Now, mice are not humans and we don't know what will happen, but those things give me reason to hope that maybe within reason we might expect to see some improvement if we can kind of remove the insult from the brain, if it's successful. So I'm not betting on, but I'm hoping that's the case, and there's good reasons to be optimistic about it, and we'll know in a couple years. Is that it?
I got one more, John.
Okay. It's like a cliffhanger.
Okay. This one's from Facebook.
Okay.
I think it's with respect to the Roche study. How obvious do the symptoms have to be considered manifest?
Yes, there's a very specific cutoff, which is diagnostic confidence level 4, which can you, in layman's terms, Mark, what does that mean?
You have to have symptoms. It doesn't matter how long. It could be, as soon as there are motor symptoms that are manifest, and the neurologist says, I'm convinced that this is due to Huntington's.
There we go. Okay. And with that, join and roll, stay informed, and I'm sorry to miss you guys at dinner tonight, and thank you for having me.